our June BHAVI symposium devoted to multiple sclerosis. We have um, 3 invited talks from Dr. Neidich, Dr. Wu, Dr. Strick, and there will also be a session by myself and Ms. Athreya on our EPS-MS trial. So with that, I'd like to turn things over to Dr. Julie Neidich at WashU St. Louis, who will give the first presentation on genetics. Thank you,
Can you
Julie.
see my slides? Carl?
Yes, everything
Yes?
looks
Okay.
good. Please.
Excellent. So I am talking about the genetics of multiple sclerosis today, which is a difficult topic, and you'll see why as I go through the discussion. I have no conflicts with regards to this presentation. And I use zebras. And so throughout the presentation, you'll see some pictures of some zebras, sometimes with some other animals. And that's because that's the way medical students are trained to consider a differential diagnosis, where the most likely diagnosis is listed first, the A, and the least likely, the Z, is the last in the herd of diagnoses. And the genetic diagnosis always was the last when I was a medical student. So we always thought of ourselves as the zebras as I went through my training time and my early facultyhood. So we still use zebras, and we'll go ahead. So my objectives are that you will understand the different modes of inheritance of human disorders. You'll learn about risk alleles and other risk factors, and comprehend the types of studies that have been done to uncover the genetics of multiple sclerosis. And within the talk, you'll note that I use sometimes the term male or female in reference to sex, and that's purely genetic sex, chromosomally assigned, and has nothing to do with gender. So first I'm going to talk a little bit about inheritance and genetics. So nuclear genes are located in specific order on chromosomes, which are packages of DNA. Chromosomes come in pairs, one from each pair inherited from each parent, and the order on the chromosomes are the same in almost everybody. And the fact is that the DNA and the chromosomes are thus present in all cells in the body except mature red blood cells. 46 chromosomes in humans, 22 are autosomes, and 1 pair of sex chromosomes. Most females have 2 X chromosomes. Most males have an X and a Y chromosome. And there's also mitochondrial genes, which are in a specific order on circular chromosomes within the mitochondria and are only inherited from the mother. And this is just a diagram where you can see that the chromosome is very tightly coiled DNA, and there may be multiple genes in any little segment of DNA, hundreds of genes along each chromosome. All of the chromosomes come in pairs, so all of the genes come in pairs, and the genes carry all the information that is necessary as a recipe for life. 1 gene from each pair comes from mom, 1 gene in each pair comes from dad. Genetics is a rapidly evolving field, and we've entered the era of multi-omics. And as you can see in this slide, we're looking at the nucleus of the cell, and inside the nucleus are the chromosomes, and then there's the structure of the chromosomes, which is called chromatin structure. There's also proteins that are bound to that chromatin structure. There's a double helix that then can be opened up. There's something called epigenomics, which is becoming increasingly used in assessing what genes are turned on or turned off. Then there's genomics itself, where you're studying the sequence of the DNA. And you can do targeted sequencing, you can do whole genome sequencing, you can do whole exome sequencing, which is the coding portion of the DNA. Or you can do transcriptomics, where you're just looking at the messenger and what is being transcribed from each of the genes. There are variants in these genes. We have something called a reference sequence, which is the average sequence that most people have. But when we have a variation from that sequence, people used to call these mutations, and they were used to designate a change in the DNA sequence. However, when you spoke with patients, we'd find that it was harmful to call their children mutants. That's what they would take away when you tell them that there was a mutation in a gene. So we're using the scientific term, which is a variant. And we all carry hundreds of variants in our DNA, which was what makes us each unique. On the other hand, there are pathogenic variants, and a pathogenic variant is one that has been shown to cause a disorder. Molecular geneticists look carefully at many details to decide if a variant is indeed pathogenic, but basically we have databases where we upload lists of everything we think is pathogenic or not so pathogenic, and we share our data. Then I'd like to discuss with you inheritance patterns. So there are several different types of inheritance patterns in Mendelian genetics. Mendel, as many people will remember, was a monk in Brno, Czech Republic, well, now Czech Republic, and he studied the peas to derive different generations of variants within those pea populations, and was the first person to talk about this kind of segregation of alleles, which are the separate variants in each of the genes. The term autosomal recessive is applied when both parents have to be silent carriers of a pathogenic variant. That means that there's a 1 in 4 chance of having an affected child. There are multiple examples of this: cystic fibrosis, Tay-Sachs disease, maple syrup urine disease, sickle cell anemia. Autosomal dominant, it means that there's a single pathogenic variant that causes the disease. If a parent has the disease, there is a 1 in 2 or 50-50 chance of passing it on to any child. There are some types of, for example, inherited Alzheimer's disease, where a parent carries a dominant variant that will give the offspring, male or female, a risk of Alzheimer's disease that's well higher than anybody else in the population. Then there are X-linked disorders. The manifestations are different in males with XY versus females in XX, with males having significantly more symptoms, and a typical X-linked disorder is Fragile X syndrome. So let's look a little bit more at autosomal recessive inheritance. Both partners are carriers of a pathogenic variant in the same gene, and that gives a 25% chance with each pregnancy of having a child affected with the disorder. If both parents are carriers of pathogenic variants in completely different disease genes, there is very little increased risk to have a child affected with either disease, although the child could be a carrier for one disease or the other, or both. Males and females are equally likely to be carriers and are equally likely to have the disorder when it's an autosomal recessive condition. And you can see in the diagram, there's a carrier father and a carrier mother, and they have a 1 in 4 chance of having a baby who gets the normal blue gene, the working gene, from each parent. They have a 2 in 4 chance, half chance, of having a child who is like them, a carrier for the disorder. And then they have a 25% chance of having a baby with the disorder. If we're talking in more depth about autosomal dominant inheritance, If one partner in a couple carries a pathogenic variant in a disease gene, there's a 50-50 chance with each pregnancy of having a child affected with the disease. That also means that the parent likely has the disorder, but not every disease has the same effect in parent and child. And I'll talk a little bit more about that in just a couple seconds. Sometimes new alterations in genes occur either in the sperm or the egg or in the fetus as it develops. These are called de novo variants. And de novo variants are more likely statistically to occur in the sperm, especially as fathers age. Males and females are equally likely to have the disorder, although for some of these diseases, males and females have different presentations. And here you see there's an affected father, an unaffected mother, and they have 50-50 chance of the father's affected gene going to their offspring, and it doesn't matter if it's the female offspring or the male offspring that is affected. Similarly, with an affected mother, you have the same pattern because it wouldn't have mattered if it was the mother or the father who carried the abnormal gene. For X-linked inheritance, if a mother carries a pathogenic in an X chromosome disease gene, there's a 25% chance with each pregnancy of both having a male child affected with an X-linked recessive disorder, so they've got to get both the XY in the forming fetus and the disease gene, which is why it's 25%. If the father carries a variant in an X chromosome disease gene, it's likely that he's affected with that disorder, and there's no increased risk to have a male child affected with the disease because he's giving his X chromosome to his daughters and his Y chromosome to his son. Thus, the son will not get the X chromosome linked disease gene from the father. Rarely there are X chromosome disease genes which only affect females. And in these families, often there are male fetuses or an increased risk of miscarriage of male fetuses who carry the abnormal gene. Lastly, there's mitochondrial inheritance, which is not Mendelian. And in mitochondrial inheritance, the mitochondrial DNA is abnormal. It's a separate circular structure within the mitochondria of cells. Only oocytes contain mitochondria that are passed to a fetus, not the part of the sperm that transmits the genome. So thus, if a mother's affected, all of her children will be affected. And if the father's affected, none of the children will be affected. There can be a mix of normal and abnormal mitochondria in every cell. So the physical manifestations of carrying a mitochondrial disorder can be very different from child to child or mother to child with these kinds of disorders. So now I'm going to talk for a couple of seconds about the genetic terminology that also will be used in this talk. And that's a complex idea that compares penetrance and expressivity, which are very different in meaning. Penetrance indicates whether a person with a genetic variant will show features of the disorder associated with that gene. If all individuals with pathogenic variants show features with the variants, when they inherit the variants with these genes, the penetrance is called 100%. So this is a statistical computational number that's arrived at through study. So let's look at a family where a child has a cleft palate and they're found to have a pathogenic variant in a gene. The father could be found to harbor that same variant but have no signs of a cleft. That indicates that the penetrance for this gene is less than 100% and maybe more like 50% to 80%. Expressivity is the degree of difference that may be found in individuals with the same genotype. That means if you take 100 people who all have the exact same pathogenic variant, how they express the disorder can be different. It's not thought of as a statistic, but rather relates to the individual variability seen in a person with a pathogenic variant. So now we need to talk about the disease in question. What is multiple sclerosis? I'm not a neurologist and I don't necessarily study multiple sclerosis. So this is my understanding. MS is a neurological condition in which there are areas of the brain and spinal cord that are damaged, causing the layer of protection around the nerves, the myelin sheath, to be destroyed. MS is considered an autoimmune disorder, which means that the immune system is attacking the body due to a malfunction. Autoimmune disorders include lupus, type 1 diabetes, and rheumatoid arthritis, among others. The contribution of genetic inheritance in autoimmune disorders is difficult to explain or tease out because there are rarely Mendelian, in other words, clearly inherited presentations in these disorders, and more often affected individuals have inherited risk alleles in a variety of genes. However, most geneticists, including myself, believe all human disease is genetic, or at least influenced by genetic or epigenetic factors. So, for example, how badly injured a person is after a car crash is influenced by genetic inheritance. If an individual has more fragile bones than other people, they may in fact have more fractures, even though they may not have a true fragile bone disease. Teasing out the genetics of common diseases like cancer, type 2 diabetes, and autoimmune disorders like multiple sclerosis or lupus has been really difficult because we don't always know the impact of a combination of genetic factors with or without any other environmental or other factor associated with the disorder. So some of the ways to work through these difficult problems are through very large studies, then meta-analyses of the very large studies taken together, and then adding in something called a GWAS. GWAS stands for Genome-Wide Association Studies, and much of the work in multiple sclerosis has been done using GWAS studies. So I want everybody to remember, though, that the Human Genome Project only published the rough sequence of the human genome 20 years ago. The complete human genome sequence was only published last summer, and there's still refinements underway in the T2T or telomere to telomere genome project. We don't know the impact of alterations in the majority of the DNA that lies between the genes, and there are a lot of regulatory elements that are still being elucidated. So we in genetics are all still learning. So let's talk about what is GWAS. In a GWAS study, a group of people, often selected because they have similar ethnic ancestry. There's a lot of GWAS studies done on the Han Chinese population. Disease process, so maybe they enroll a lot of people who have multiple sclerosis or another disorder, or just because they sign up and they all undergo testing for a specific set of DNA locations or single nucleotide variants, SNVs. There are large data banks of genome-wide associations that can be data mined. So there's the UK Biobank, there's the All of Us study, which is across the US. There's certain medical systems like Geisinger, where every patient at Geisinger has the opportunity to have genome sequencing done. And then there's the Kaiser system in California, where again, they have a large database of people who have just been sequenced. The other point here is that the SNVs, or the single nucleotide variants, are often selected because the lab has the right reagents to test them. They're included on a already set up microarray that the lab has, or because there were prior studies that showed these specific variants were useful in those other studies. Test is usually run as a microarray although DNA sequencing may be used instead. And the analysis of the data will show if the SNVs in the disease process are both found in the same individuals. So you have a bunch of single nucleotide variants and a bunch of people who have the same disease. And if these match up, then you've got an association. GWAS results don't necessarily show specific correlation of a gene or a variant and a disease process, just a statistical association in the tested population, which is a very important concept and sort of hard to grasp. You sort of think, oh, they ran a GWAS study and found this specific SNV associated with multiple sclerosis. That doesn't have to be the disease allele. This could be just located in a lot of people who wind up having the disease. So there's a lot of thought that has to go into the interpretation of GWAS studies. So what does that mean for MS? Since MS is not usually inherited as a Mendelian disorder, there are risk factors that can be inherited, and as well, there are environmental and other factors that influence whether a person develops MS. Most gene variants that increase the risk of MS have been identified by GWAS studies. Very few families with an inherited form of multiple sclerosis have been identified, and only a portion of those families have undergone sequencing studies. And basically, you would need probably 3 or 4 affected people in the family to be able to tease out what is inherited along with the risk for multiple sclerosis. And then there may be skip generations and other issues that make this analysis particularly difficult. And then finally, there are many other factors that are associated with MS, including a potential association with later onset infection with Epstein-Barr virus, EBV. And that's been in multiple papers in the past couple of years. So we're looking now at a diagram, a graph that explains a little bit about how people gauge allele frequency, which is how frequently we find a specific SNV or other genetic variant in the population. Penetrance, which again is that percentage of people who actually have the disorder when they've inherited a gene for a disorder, and disease. So if we look at this, we can see that there are some very rare variants that are often associated with Mendelian disease, and they have high penetrance. In other words, most of the people who carry them will show signs of the disease. There are some very rare variants that are hard to identify genetically that pop up on all of our sequencing assays, and it's hard to associate them with disease. There are low frequency variants that have intermediate penetrance that are relatively uncommon, but may be associated with disease. Then there are variants that we all carry that are highly unusual in common diseases. And then there's a large number of variants that have low penetrance that are identified by GWAS studies. And those are usually common variants, which is another reason why interpretation of GWAS studies is so very difficult. So here we have some zebras and giraffes, and this is what we geneticists are always looking for, the zebras. Recent research. So there has been research to create risk assessment formulas that take out data sets and assign risk to factors that are most likely to be combined in patients who are diagnosed with MS. And so you might take, for example, the age of the patient plus the genetic sex of the patient plus a score that is derived from the presence of several specific genetic variants. And that may allow clinicians to better figure out which individuals have a greater risk of developing MS. Whether the members of the family have MS is often part of the risk equation, as well as some other polygenic risk calculations. So usually there's one set of data that's used to figure out the formula that's used to create this risk assessment, and a second separate data set that's used to test whether the formula works. So you need a lot of data in order to do this kind of risk assessment formula. This is a graph from, or a table, it's a workflow really, from a recent article by Lou, where he was, this group was putting together a risk assessment. And this included sequencing studies, and there was a host of other information. So you see there's GWAS data that's been included, gene expression studies, DNA methylation studies, which have to do with epigenetic factors. There are other different kinds of sequencing studies. There's a lot of gene risk validation. There's candidate genes in here that are being included. So they do a lot of work to arrive at something that they think is significant and will then help predict whether or not a person is at risk for MS. This is a separate study that was a workflow from a different group where they took GWAS data and they basically had a huge amount of data. There were 14,000 plus cases, 26,000 controls. They looked at all of that data for heritability and sparsity, the rareness of a disorder, and they looked at the validation and test sets, and they came up with a score that they then validated using data from the UK Biobank, where there were 100,000 controls and 600 cases. Then they tested with a separate group from the UK Biobank, so that's phase 2, different cohort of people, and also in the Kaiser Permanente Northern California cohort, which included cases and controls. And so finding the right variant is hard, and it's just like looking at a Where's Waldo. In this, Waldo is up in the upper left-hand corner, but there are other individuals who have red stripes, and perhaps everything with a red stripe increases your risk of MS. And the only way to analyze for this is to pull in all the data, use bioinformatics, and then analyze the statistical significance of everything you see. So, what factors were found to increase risk? So there were a lot of genetic factors that were included in this. There were pathway-specific factors and genes in adaptive immune responses, that was IL-5 and IL-2 signaling, T-cell receptor signaling, MHC class II antigen presentation, interferon gamma signaling, complement cascade genes, viral and parasitic infection response pathways, pathways identified in other autoimmune diseases like lupus, Hashimoto thyroiditis, or type 1 diabetes, cell adhesion and extracellular matrix organization, protein glycosylation genes, and the VEGF and Notch pathways. There also was an associated risk in chromosome 6 genes, where there are several immune response genes located. So chromosome 6 has a lot of genes that are called HLA, and those definitely have to do with how our bodies react to foreign substances, etc. Now, I wanted to just touch on this because there was a lot of talk about EBV infection. And there were many papers out in 2022, just in the past year, that looked at large data sets. For example, military recruits who were tested for exposure to EBV, or data from countries where the medical system regularly tests the whole population. And actually, I think the data came mostly from Denmark. And there were arguments about the quality of the analyses. There was actually somebody who published, or a group that published a paper saying, oh, you didn't get this, you missed something, you've completely misunderstood your own data. And then other people who came back and said, wait a second, no, we really did understand, you don't understand. So competing papers about this particular association. And some of the analyses suggested that individuals infected with EBV for the first time as young adults may have increased risk of developing MS. And most of these studies did not test the participants for genomic data. So there isn't a good list of genes or variants that accompanied those papers. And so there's very little that can be done except taken as a statistical analysis. There was no data that showed that EBV alone caused MS in every infected individual. And I think that there could be a specific set of genetic variants that allowed people with later onset EBV infections to then go on and develop MS. We don't know. And I hope that the people who published these large data sets about EBV will also start looking at the genetic contribution in their patients who either did not, or their subjects, I should say, who did not develop MS, or the subjects who did develop MS. This is something that's going to be probably debated for several more years. But there is evidence that there's statistically a risk factor. There have been papers published in the past looking at the risk of other childhood onset disorders like rubella or measles, and none of those panned out. So there was not an association of MS with having had one of those childhood diseases, which we mostly are able to prevent these days due to immunizations. So looking for zebras amongst the herd. You're going to find a lot of variants when you start doing genomic sequencing, or even looking at the GWAS type data that's usually done off from a microarray. You're mostly going to find benign variants that are or commonly found in populations just because the individuals in the population are usually from the same geoethnic ancestry. In other words, if you take a lot of people who have the same genetic backbone, they're going to probably have the same variants. The question is, which of those then are associated with increasing risk? And that's where I think that we don't have a really good handle yet, but the people who are looking at the data, including the multi-omic data, to try and derive a score are probably the people who are more likely to figure out some kind of a specific risk assessment to do when we're looking at people for a genetic or other reason for risk of MS. And in summary, to understand the different modes of inheritance of human disorders, we've gone through a discussion of Mendelian disorders, mitochondrial disorders, and the fact that MS does not follow these modes of inheritance. We talked about other risk alleles and factors like EBV infection. And we've talked about the kinds of studies that have been done to uncover the genetics of multiple sclerosis, from looking for risk alleles using GWAS studies, families with several affected family members who have been sequenced, and then scientists who are then analyzing the data along with other multi-omic data to derive polygenic multifactorial risk scores. Polygenic implies solely genetic risk. Multifactorial takes genetic risk and other factors. I think that we're probably looking at a multifactorial risk as opposed to a sole genetic risk, but I do believe that there are risk alleles and genes that will be identified. Thank you.
Thank you very
Are
much,
there any
Dr. Neidich.
questions? Yeah.
Yes, I certainly have questions, but anybody else want to ask their questions first? Well, let me ask some of my questions. So with regard to the statements about non-association or no clear evidence for association um, with some common childhood diseases, uh, including measles, what about association if the individual um, experiences presence of the clinical disease as an adult? So what about association with measles, not as a child, but as an adult? Is there any evidence for that?
You know, I didn't find a lot of papers looking at late exposure for those kinds of infections. There may well be, but you know, it was not easy to find. I only found some data about early exposure. Um, Maybe Dr. Wu has some thoughts about this as somebody who sees a lot of patients who have MS.
I will cover a slight bit of this too in my talk, but one key ingredient is that EBV is a latent virus. And so we're all exposed to many viruses that could push the immune system to a direction or a response. But the idea that the virus sits over time to create a sort of snowball effect may play a role in this as well.
Yeah, indeed. And you know, there even are people who have genetic variants that cause them to have, say, specific types of cancers after EBV infection. So EBV is well known to stay around, stay latent, and not necessarily be cleared out. Similarly, hepatitis viruses are like that, although I've never heard of a specific association with MS.
And then, of course, the other associated question relevant is the occurrence of the viral disease, of the viral infection, contemporaneous with pregnancy. And we know that there must be alterations of the maternal immune system in order to, shall we say, host the pregnancy. So what about associations when the viral disease occurs during the pregnancy of the mother, who then experiences multiple sclerosis a couple years later, 5 years later?
I think this is hard to tease out. And part of the reason it's hard to tease out is because most people these days are, in fact, immunized and not, there just isn't a large cohort of individuals at this point who could be followed out who have actually had those childhood infections. Clearly, COVID is a different example, but nobody has found yet an association between a SARS virus and multiple sclerosis. There are clearly, though, some people who have long-term neurological effects of having SARS-CoV-2, which is the COVID-19 virus. I think that this means that there's lots of work still to do.
Yeah, well, I think these questions have to be addressed independently of each other. So, a 22, 24, 26-year-old pregnant mother may suffer a viral illness that has nothing to do with her past history of, infections as a 3-year-old. So this may be completely independent of whatever diseases the mid-20s mother may or may not have had as a child. So
Sure.
we have
The
to
problem,
separate the question of past history in the 20-something-year-old mother from the question of the infectious illness simultaneous with the pregnancy. I think
absolutely, Carl.
I actually believe it's an important model to study the immune system because of
Carl,
the complexity of the questions involved related to pregnancy.
Carl,
It-
absolutely true. But what I'd like to say, though, is that if you do not have people who are exposed to those disorders, those infectious diseases at this point, finding a cohort to study forward to see who then develops MS is hard. So the some of the work has been done, for example, in taking recruits into the military, where they can be studied serially for whether or not they've acquired EBV or other infections. I'm sure that they get studied for CMV and a whole lot of other viruses.
I,
The, the,
to make a joke, which probably is not politically correct, I suppose one would have to consider the anti-vaxxer cohort.
well, there's them, but at the same time, there still isn't raving infection in the US, at least, of those childhood diseases in adult people because all of the anti vaxxers were all vaccinated as kids, right?
Right.
We have to wait to see another 20 years when their children who have not been vaccinated develop these.
Okay, a completely unrelated question, which will, I believe, be the last question unless anybody else has any other questions. Can you comment? I was curious about the FAIR sequencing method. The spelling with the acronym FAIRE, and I understand that there are a variety of different sequencing methods. Can you just comment briefly? Which are the most commonly used, the most routinely used in the genetics labs these days for sequencing in the clinical setting? Is
Yeah,
which
not that.
methodology? Okay, so
That
which?
It's regular NGS, next generation sequencing, typically done on some of the platforms from Illumina or from Thermo Fisher. There are other companies that have other similar sequencing methodologies. We at Washington University use the Illumina platform for all of our next generation sequencing constitutional studies and also for our cancer studies clinically. And there is a clinical lab that has all of the typical regulatory elements, CAP and CLIA included, for doing these kinds of sequencing assays in patients.
So that FAIR method is not really used except in research context.
Right.
Rarely used then.
Right. And it has nothing to do with any other kind of FAIR that one might have heard of.
Okay. All right. Any other questions? Yeah, before I stop the recording and we take a break until the start of our next talk by Dr. Wu. Okay, we'll reconvene in 15 minutes at 1 o'clock California time. Thank you, everybody.